Okay, hello everyone. My name is Alex, and I'm the founder and CEO of the Singularity Squad Association. Singularity Squad is a youth longevity association with a mission to popularize longevity among young individuals worldwide. Longevity in general is not just a scientific concept, but also a fascinating field that holds the promise of extending human lifespan. Just imagine having an opportunity to live as much as we dare to dream. Our main goal is to spark a curiosity and inspire youth to explore boundless opportunities that longevity presents by organizing events, having discussions, and providing various resources for young people to use. For example, recently we have collaborated with Open Longevity and participated in the Save Forever movement. Our members went on the streets in Florida and engaged in conversations with people about longevity. Also, every week we meet online through Zoom to discuss interesting topics and discoveries made in this fascinating field. As for resources, we regularly post interesting articles on our website and on our Instagram. Each of you can help us in accomplishing our mission by participating in our events, our weekly discussions, or by just simply reading our posts and articles and share them with your family and friends. Now I want to ask my co-CEO Fernanda to talk more about our international chapters.
Hello everyone, my name is Fernanda and I'm the co-CEO and representative of our Chilean chapter, and I'm thrilled to share the excitement that our events in Chilean schools have generated. We held several presentations in different high schools across Chile. We talked about our association and longevity, and it was invigorating to see the genuine interest among students. We had dozens of students reaching out to us and joining our team. Singularity Squad is an international association and currently has 11 chapters in 9 different countries, such as the USA, Chile, China, Japan, Ukraine, Turkey, Azerbaijan, and Germany. No matter where you are and who you are and what language you speak, you can all join our big international family. And now Alexander, the representative of our Russian branch, is going to talk about events held in Russia and our research opportunities.
Hello, do you hear me? Hello, everyone. My name is Alexander, and I'm a representative of Singularity Squad in Russia. In September, we had a privilege to present at one of the most prestigious Russian universities, the Moscow State University. And one thing that I like about our presentation is that the cricket transformed into interactive discussions with audience. It's so interesting to hear people's different ideas and perspectives. However, besides presentation, Singularity Squad offers opportunities for young people. And if you have an idea for longevity research, please reach to us. We can assist you with mentorship, university partnership, and funding opportunities to turn your longevity research projects into reality. So just simply go to our website and click to button research, select and fill out a quick form. And thank you for attention. And now please welcome the scientist, Peter Lidsky, the lecture evaluation of egging in search of a progeny.
Thank you very much for this invitation. Thank you very much for you for coming. So today we will speak about a little bit broad topic. Can you see my screen now?
Yeah, confirm.
Yes. So the question is why? The old age, nobody understands what for. This is a little bit sad and strange, but if we ask different scientists, why do we age? We find lack of consensus. So the different people surprisingly give us different answers. And this is a paper that has been published like a couple of years ago, and a lot of scientists gathered together to find a paradigm of what they call aging, and they agreed to disagree in the end. Everybody came with their own opinion and left with their own. And why is it important to understand? Because generally, one can say, one can say that it's not very important. We need to find out druggable targets, test drugs that prolong lifespan, and the problem will be solved. Why do we need to understand the nature of the event? So let's start with something completely different, not related even to biology, just to emphasize the value of the concept of a paradigm in any scientific research. This is the first computer that has been ever done by humanity, to our knowledge, Antikythera mechanism. It was found in a shipwreck. So the date of creation is about the second year BC. So, and it was found as like a box with rusty gears. And then some of the archaeologists came up with the idea that this is a calendar, something that predicts the solar eclipses, Olympic Games. And based on that model, now we have a full reconstruction of the device with certain moving parts. So 1/3 of parts were not were missing entirely, but we understand how it was working to very high precision. And let's say we wouldn't have this idea about that this is a calendar. We don't know what was the input, what was the output, and what process the mechanism should mediate. Then what would be our approach to figure out what it is? Like to trace back every molecule of this rusty brass and track back to its real position. And like this sounds like a very complex physical work, and we will probably learn a lot along this line, along these lines about how the sea currents are affecting the decay of brass, how the sea organisms are affecting the decay of brass, but we will probably never have the full model here. So that's why I think the idea of the purpose is essential. And now we come to aging. So the hallmarks of aging is another sort of paradigmatic piece that is persisting in the field. So it looks like that. I endorse you to read this paper. So just this paradigm like consists of 12 elements. I believe it's now 12. And like everything that people are studying in aging is listed in this cake-like structure. So like dysbiosis, genome instability, telomere attrition, epigenetic alterations. So it is not a paradigm and the idea of the purpose. It's more like a catalog of the things that people study. And question is, what is a primary reason of aging and what is the secondary reason of aging at this level? Because let's say it might be that some of these effects are secondary. And to explain this idea better, I want to deliver you the problem of glasses. This is a favorite Vadim Zgladyshev example. So let's say we have old people, they get myopic, and they got hit by cars and die. So now we give them glasses. With glasses, we improve their quality of life. We increase their survival because they are not hit by cars anymore. But with this, we don't cure aging. We just deal with one of the symptoms of aging. And same thing can happen on biochemical level. So we can do something, let's say, about wrinkles, but it will not help on systemic aging. And the researchers will be used not very efficiently because the progress will be minor. That's why I believe we must know what is the primary reason for aging. And to do this, we use evolutionary biology. This is a famous evolutionary biologist, Theodor Dobzhansky, who said nothing in biology makes sense except in the light of evolution. And this is very true because our bodies are made to propagate our genes. And this is the only thing we are really sure about biology, that biologically we exist to replicate. And now, of course, aging is a little bit strange phenomenon in this paradigm, because if I die due to aging, I cannot replicate anymore. And how to put aging into the evolutionary paradigm is a big conundrum. And what ideas has been proposed so far? So the first relevant idea was produced by these two scientists. The great Medavar was an immunologist, Haldane was an evolutionary biologist. So the selection shadow. That idea is that animals are dying of extrinsic factors like predation, cold, hunger, infectious diseases, and with every age, so less and less animals are surviving, right? So if no one is surviving to the age of 25 years, then it doesn't matter for evolution what happens after this cutoff. And this is called selection shadow. The evolution doesn't care what happens after a certain age. However, if we look into real data, this is, for example, for lions, so we find that animals are aging in the wild. So that a lion of age 14, here is mortality rate, here is age. The lion of the age of 14 has much higher probability of dying than the lion of age of 5. And lions survive to age of 14 in the wild. So the selection shadow clearly can work, but it is not sufficient to explain aging, because it requires this law not to exist to work. So here, if we will get a mutant lion that would age less, this lion will get opportunity and to propagate its genes better. So the next model that has been proposed to resolve this problem was proposed by George Williams. And his idea was that there are some genes of special sort that are good in young years and bad in old years. And so, because in very few animals survive to old years, so this can indeed work. So the individuals who are having these genes are so strong and robust in young years that they leave so much progeny that they die later, a little bit earlier, but nobody really cares because the progeny is already propagated. So this model can work on the paper. However, it was proposed in 1957 when people have little idea on molecular genetics. But for looking from now, it requires non-evolvable features. Let's say lifespan of a rat is 2 years, lifespan of a squirrel is 20 years. The two animals are very similar relatives. So does it mean that genes that mediate aging in rat are different from genes that mediated aging in squirrel? What is the reason why rats cannot evolve longer lifespan than squirrel, propagate their genes for longer, take advantage of this long lifespan? So this this question is not resolved by antagonistic priority. So it seems to be there is a certain ecological trade-off between aging and some adaptive trait. And one of the most popular models of this trade-off has been proposed by Tom Kirkwood in 80s, 90s, 70s, 80s. So he came up with his disposable soma model that we invest in reproduction. So we have a limited budget of energy. We invest in reproduction at the expense of repair. So let's say in this example, this is aging host. It reproduces extensively because everything is put in childbirth. And this host is investing in repair, and it replicates less. So by time 2t, this host will have 3 individuals. This host will have 4 individuals. And with this, because this is exponent, so the the ager can outgrow the non-ager. The idea is compelling. However, it has some, first of all, it has some mathematical problems because it indeed can work in exponentially growing populations. However, if we have an exponentially growing population, it means that there is a lot of food around. And if because to grow exponentially, we need to feed all this growing progeny. But it is a lot of food, but at the same time, it should be not enough food for the parent to maintain their bodies, right? So there is a contradiction on this food availability condition. And very few populations in nature are constantly growing exponentially. Mostly they oscillate around certain optimum. And in stagnant population, like most of the species are existing in, so it can work only if producing new individual is cheaper than maintaining the parents. So it would mean that like I need to heal my body, right, and all the nutrients that are invested in that, and make another Peter from a fertilized egg, which I think it cannot be like economically advantageous anyhow, because I lose all my nutrients. I think the claiming that this can be a saving strategy is a contradiction to the basic laws of conservation. In addition to these arguments, disposable soma also have specific problems like caloric restriction. If we feed animals less, they live longer, while the model predicts they should live shorter because they don't have enough resources. So males are aging faster than females. Males are investing much less in reproduction. Food consumption decreases with age, while from the model it should be other way around. The older individuals should eat more and repair better. In poor ecosystems, like in Arctics, in the troglobionts, all the deep water fauna, they are all enriched with long-lived species. So under poor conditions, long life is favored. So it is very unlikely that aging evolved as an energy-saving strategy. So this antagonistic pleiotropy and disposable soma, they are often summarized as pay later models. So we borrow our fitness from our old years to be strong and robust when we are young, and then we need to pay back this debt. This later, this formula, it excludes rejuvenation, because if rejuvenation would be possible, evolution should make it all the time. We cannot imagine the bank loans in the world that have a time machine, because people who are to pay their interest to the bank would go back with the time machine and pay their debt without interest. So if we borrow our fitness, and the idea that we must pay the interest, if rejuvenation is possible, then it should happen every time we approach old ages. So if evolution haven't done it, then rejuvenation is impossible. Very bad news for us if we believe these models. However, good news are that rejuvenation is possible, and it can be observed in some animals. So specifically in the eusocial bees. You know, eusociality is when we have a queen and workers that do not reproduce. Like that's a social structure of the hive. Only the queen lays the eggs, and also in this hive, the bees, they have like a age stratified jobs. So then the bee is hatching. First, it serves as a nurse, taking care about eggs and larvae and pupae, and then it goes to foraging job to collect food. This job transition is accompanied by a dramatic decline in health, and then after a couple of weeks, this bee dies. However, if we take a hive, we toss all the nurses, then some of these foragers need to go back and take their jobs. Otherwise, the hive will die. So, and then they rejuvenate and they live much longer. And they, by health metrics, they are healthier. They are younger. So, and this shows that first, rejuvenation is mechanistically possible. Second, it occurs under the stress and not under normal conditions. And this is a strong contradiction to the pay later model. Next, there are other non-explained phenomena that classical models cannot explain. Sorry for bad phrase sentence. So, for example, everything that flies lives very long. So this is body size. This is maximum age for mammals and flying birds. And as you can see, flying birds are living relatively longer than mammals of their sizes. Next, bats, flying mammals, are also living longer than the terrestrial mammals of their sizes. It seemed to be that it is about flight and not about the accompanied phylogenetic components, because flightless birds, like ostriches, penguins, they live relatively shorter than flying birds. So it seemed to be that this is more about flight rather than about being a bird. And this is not explained by classical models. Next, there is this similparity, which is not aging itself, but it is sort of a programmed death. So you know the salmon fish that go up the river, lay eggs, and die, like within 2 weeks from accelerated aging. There are octopuses that do the same. They don't go up the rivers, of course, but they die after laying eggs. And there are also several mammals, and all these mammals are carnivorous marsupials. From these two are from Australia. This one is from South America, that also do that. And in this clade, this similparity evolved several times, 5 times independently. So in most of these species, just males are dying after reproduction. In this animal, also females are dying after they accomplish weaning, and surprisingly, males. So in these 2 species, males die soon after they mate. In this species, males are dying together with females after the females accomplish weaning, which is a little bit bizarre, and because the classical idea about similparity based on disposable soma model is that these animals are exhausted by mating or weaning. But in this case, it seems to be that the males have had time to feed, but they somehow don't use the opportunity. and what for they dying is not explained. So also, if we speak about eusocial animals, like you remember, queens and workers that do not reproduce, this eusociality is around in some insects, in termites, bees, and ants, and also in some mammals. So some rodents are also eusocial. This they have breeders, the queens, and non-breeders. And in both insects and mammals, eusocial queens are living way longer than workers, having the same genome, right? Why? So there should be two different programs for programs of aging within one species. Why does it happen? The classic model cannot explain that. And there is another example that this animal is also eusocial. So they also have queens and workers, but in naked mole rats, both queens and workers basically do not age or age very, very slowly. And this is not clear why the animal the size of a mouse can live for 30 years. Homo sapiens, our species, and these naked mole rats are two clear outliers from the general array of mammals. And the classical model cannot explain the ecological reasons for that. So now let's step back and think what model we need, because there are about 300 models on the table, and how to select and how to sort them. So I think that first what we need to ask the model, can the maladaptive effect be mitigated with energy investment? For example, one of the models says that, okay, we all breathe, so this oxidative phosphorylation is very good, but it produces reactive oxygen species. Maybe we want, maybe we age about because of that. But then we just can synthesize more catalase, an enzyme that destroys reactive oxygen species, and with this we can overcome this maladaptive effect of oxidative phosphorylation. So if we can solve the problem with energy investment, then it is disposable soma, and we know that disposable soma isn't good. Next, can the model explain somehow the differences in lifespan between relative species? For example, there is a model that mitochondria, the power machines in our cells, they evolve because they have their own genomes. They evolve, and they like non-functional mitochondria that reproduce very, very fast can accumulate, and this can result in aging. However, this process probably indeed exists. However, mitochondria are in both rats and squirrels, and the difference in lifespan is tenfold. So it cannot be just mitochondria. So if we cannot explain differences between different species, then it's a poor model. And in addition, if we can explain that, the ideal model that we want to have should explain all these exceptions and peculiarities. So today I will present you an attempt of building such a model, and I think that according to this criteria, our model is the only one that fulfill them. And question, high-level question: can aging be programmed and adaptive? And traditional answer is no. And first argument that traditional gerontologists will provide to you is that Hamilton's rule that tells about evolution of altruism. So C is the cost for the individual who produces the altruism. So like how much I give. B is the benefit for the recipient, and R is relatedness. So for altruism to be evolutionary stable, evolutionary advantageous, we need to help our relatives, right? So let's say if I have an ice cream, I give it to my twin brother, the same genome. So then I'm in 0. If I give my ice cream to my brother, just sibling, then I lose 50% of this ice cream because my brother shares only 50% of my genome. Right? So the altruism can be possible if I have 10 ice creams, they are melting, and I don't know what to do. I cannot eat them. I don't know what to do with them. I give them to my relatives. Then altruism can work because cost of these ice creams for me is low, benefit for them is high, right? But in case of aging, I kill myself. I give away my life. How can it be possible that this benefit is so huge that it is outgrow my the enormous fee I pay? It seems to be impossible. Second argument against a programmed and adaptive aging is where are non-aging mutants? Okay, let's say aging is a program. It's somewhere in our genes. There is a gene of aging. We can take our flies or worms or other model animals. You can mutagenize them. Then you need to find a non-aging mutant. Or there are like some billions of people on Earth. Right? It's also a good large genetic screen, but we never see a non-aging human. Well, we can see humans with all the possible defects. We see something that we can interpret as a gain of function phenotype, but we never see this immortal loss of function phenotype. So that's most of the gerontologists do not believe aging can be programmed and adaptive. So now, let's speak about the model we developed. So the basic idea is very, very simple. We have chronic infections around. And this is example of this fish and this parasite. With the age, the rate of chronic infections is increasing. If the infection cannot be cleared by immune system, if the probability of infection, let's say per year, is same, then at some point, after a certain age, all the individuals will be infected. This parasite doesn't sterilize fish. Fish still can reproduce with this parasite. But let's say this parasite is sterilizing. Then, after a certain age, none of these fish would be able to reproduce, and they will swim around and spread parasites to their relatives. And this fish can be a problem for its genes rather than a benefit, because it doesn't spread the genes. It harms other fish of the similar kin, similar gene makeup. Of course, we are not able to run evolutionary experiments in nature because it takes too much time. So we at this stage we mostly use computational models, and we build these models of viscous populations. So like animals are moving on the surface, like they are territorial. They reject each other like charged balls of the same electric charge, and you can see these tracks that remind us the GPS tracked wolves in Minnesota, like a real example of territorial animals. So idea of this territoriality is that they maximize the area they control, and the resources that they use to reproduce are dependent on this area. So this model recapitulates two important factors that are typically missed in the epidemiological modeling. So first, there is density-dependent dispersal. Animals from dense areas are moving to the more sparse areas in search for food and other resources. So this is a very conserved pattern of behavior. We can find it in bacteria. It's called quorum sensing. And second, there is some gradient of related. So the relatives are interacting with their relatives more often than with just random members of the population. So like interaction between brothers or cousins happens more often than just random animals. So, and how does epidemics run in this population? Let's say this is these yellow dots are short-lived hosts, red dots are long-lived hosts, and now the red rings are pathogens. And you can see that, and this is a sterilizing pathogen. So you can see that in short-lived population, the epidemics is there, but it's not that dramatic. But in long-lived population, it is very high because they these guys are dying after 90 time units, and many of the infected guys are dying, and these guys are surviving. So just to make sure we are on the same page, pathogen doesn't shorten the lifespan, because generally chronic pathogens do not want to do that. They want hosts to live as long as possible to spread. So now, if we compare these two strains, so here is a short-lived population. Here we put in a long-lived variant, and the long-lived variant is taking over because they live longer and they propagate better, right? And this is in the absence of disease. Now, what happens if we add the disease into the system? Then, also in the first moment, this long-lived variant is expanding. Then it encounters the pathogens, and then the pathogens are sterilizing. So the epidemics is much stronger in this long-lived variants. The pathogens are expanding there, sterilizing it, and then the demographic pressures from these short-lived these areas of inhabited by short-lived animals are displacing the long-lived. So, and therefore, this pathogen pressure can control and prevent the emergence of longer lifespan evolution. And with this, we can resolve the Hamilton's rule problem. So, because here the cost is declining, because if I'm sterilized by the pathogen, then my life doesn't cost much. And second, in the left part, we have a sum, because by killing myself, I can help multiple individuals to stay healthy and propagate. And this modification to the formula can resolve the argument number 1 against adaptive phage. And what about argument number 2? Why don't we see this Voldemort mutant? So the answer, we believe, lays in the unusual selection dynamics, because the long lifespan in the first time points, in the short run, is advantageous. So it is good for propagation. Then something happens, namely the pathogen is encountered, and then this property becomes disadaptive. So the same trait is adaptive at first and disadaptive later. This is a very unusual situation. And the color is wrong. Let's run another experiment. Here we have 3 different strains. Yellow ones are short-lived and cannot make a long-lived mutant. Brownish ones are also short-lived, but they can make a long-lived mutant. And red one, this color should be red, are just a long-lived mutant. Then let's see what happens. So look at this part of the plot. So we see this immortal mutant is emerging. It's displacing its paternal brownish, and then it encounters pathogens and is displaced. So with this with this experiment, we show that not only being long-lived, but even being able to produce a long-lived mutant might be disadvantageous. So that we, if we can produce a long-lived mutant, it will first displace us and then die by itself. And the main benefit will get our neighbors, our far neighbors, that cannot produce this mutant. And to summarize, if we can go from aging state to non-aging state in one mutation, it's a big problem. So how evolution can resolve it? We probably want to have a multigenic control of this, that if we have it if you require 2 mutations to go there, then it is rare. It's very unlikely that 2 mutations will occur in the same individual random. However, let's say one of these genes can get mutated, right? Then this intermediate form that still can age can accumulate in population and then go back to this unwanted long liver. And our model says that evolution wants to attenuate these intermediates, that they don't accumulate. And this there are some evidence for pleiotropic genes sort of around that if we do a long-lived mutant, it is compromised because of something else. So some other disadaptation. They are either less fertile or less stress resistant or weak and sick. So, and we believe that real explanation for that is that the evolution wants them to be attenuated, that they do not produce really biologically immortal individual. So with our model so far, we can we fulfill this part of we don't need energy investment. We can explain the differences between species by different population structures, different pathogen dynamics, and we also resolve the problems of the adaptive aging, the Hamilton's rule, and this non-aging mutant problem. Now, what about these peculiarities that these outliers that cannot be explained by classic models? Let's try to resolve these. First, we go with the flight. With the flying animals have 10 times higher dispersal than non-flying animals of their sizes. So, and if we, as you might remember, viscosity is a key parameter in our modeling. So the individuals should interact with each other like kin specifically. So relatives should interact with each other more. So if I want to kill myself to save everyone on this Zoom, it will not help my genes to propagate. I need to sacrifice for helping my own kids or siblings, right? So if this is called the tragedy of the commons in the game theory. So if we don't have viscosity in the model, then long life wins over under any conditions. So in the presence of the pathogen, in the absence of the pathogen, and independently on the pathogen properties, just the whole population gets infected. Everybody is very unhappy, but these altruists that are short-lived, they already are all in the grave. They get extinct, and we believe that this viscosity is an explanation for association between aging and lifespan and flight. Next, if we speak about the eusociality, the queens consist a small proportion of the colony, and their long lifespan cannot impose a large epidemiological risk on these populations. So the main control for pathogens is mediated by the short lives of workers that are the majority. Yes. So now, what about the naked mole rat? I want to remind you, both workers and queens are not aging in naked mole rats, or aging very, very slowly. So our hypothesis is that these animals are hypersensitive to infection. So this is also an adaptation, probably possible only in under eusocial population structure. So if the very first animal kills itself in response to any infection, then the colony can be kept basically free of pathogens. So we don't age. We just kill ourselves immediately if we feel we are infected. Then we control pathogens very well. Then aging can be redundant. If aging is really a pathogen control mechanism. And indeed, there are papers case reports saying that mole rats are indeed hypersensitive to certain viruses. So this should be studied in more detail, but I think the data we see now is pointing this direction. Now, also the question, why is rejuvenation so bad? So why these bees are rejuvenating only under the threat of death of the meta-organism? Where this choice between the rejuvenation or death comes from? Why we cannot use rejuvenation routinely every time the forager reaches its old age? So the pathogen control gives us a rationale, so that this forager has a high probability to be infected with something bad, and it needs to die. And regularly, we want to remove it just for the prophylactics of infectious diseases. But of course, if we are running out of nurses, we must bring him back and use regardless the epidemiological risk, because otherwise all the larvae will die. So, and a little bit consistently with this, some pathogens can prolong lifespan in eusocial insects. So this is an amazing paper coming out several years ago, 2 years ago. So there are ants, also as in all eusocial insects, the queens are living way longer than workers. But if workers are infected with tapeworms of certain species, then these workers start to live as long as queens. The difference is dramatic. So, but it seems to be that parasite can switch the program. And of course, for the parasite, death of these worker ants is very, very bad, because it will not be able to propagate. And like that, it can extend its infectious time. So what about similparity? So we believe that similparity evolved as a method to prevent transgenerational pathogen transmission. So the parents die together with their parasites, and with this they save the progeny from being infected. So so we think that in case of Pacific salmons, it's their population structure, that they swarm like hell during these spawning times. So for octopuses, we don't have a bright idea. For marsupials, we have explanation why, what is unique about their immunology and the population structure. But this, I think, I will take care of time. I will maybe tell next time. And now I'm finishing and happy to take questions. Any questions, guys?
I'm still trying to think of one.
Thank you. Yeah, really exciting.
Yeah, guys, don't be shy. So we don't expect everyone here to have like a strong biological background, so there is no stupid question to ask. So yeah, please don't be shy and ask something. I will also think of something myself.
I would like to ask something, if I may.
Sure.
I will skip the introduction, who I am and my background. I'm a biologist. Have you considered plotting in any of these evaluated species sexually transmitted diseases, or how would they impact?
Yes. So the sexually transmitted diseases are key for of course. So I think in population of mammals, these are probably the main factor, because some of the sexually transmitted diseases use sterilization as their part of their reproductive strategy. Because if a female cannot conceive, it starts to try different males, and this improves the Timothy, you have a party in the background. So most of the sterilizing diseases of humans, let's say, are sexually transmitted, like gonorrhea or syphilis. So and HIV also sterilizes to like 40%. So yes, this is absolutely a key factor indeed.
Interesting. How would that play out for, say, for naked mole rats that already have hypersensitivity to numerous factors, such as hypersensitivity to contact inhibition and thus cancer resistance? And now, after what you described, hypersensitivity to the infectivity or the ability to get infected with HSV1, with herpes simplex virus 1. So yeah,
just to make sure, in this case hypersensitivity was that they all died. So they are not they are probably infectable as mice. So because this herpes is used to deliver just as a vector, to deliver genes into mice, but mice don't care about being infected with this virus. Yeah. But all the mole rats died in this experiment. But of course, this is only one N, so one need to test other viruses on naked mole rats, which is difficult because these animals are so precious. No,
so perhaps go ahead, sorry.
No, I think sexually transmitted diseases are not very important factor for naked mole rats because they are probably they are predicted to be pretty much pathogen free.
There's also a question in the chat from YouTube. Danilo posted it into our internal chat. So great talk as usual, Peter. You believe pathogen control is one reason why agent was selected for, or are you arguing that pathogen control is the only reason agent has been selected for? Also, you believe that the body has a clock and knows how old it is. What progress has been made in finding this clock?
Okay, thank you very much for these questions. So the first question was, is it one of the reasons or one or the only one? I believe that if we remove pathogens from the system, then most of the species will evolve towards non-aging. So still there are examples where clearly this will not happen, like killiefishes, like that live in the ponds that dry out during the summer. So there, with the species with deterministic death due to non-pathogen reasons, non-aging will not work. In all the others, I expect aging to be lost, The second question was the clock. Yes, we have a clock. I believe in that, and so just visually we can judge how old the person is with a high precision. So it's clear that our body is measuring time and like evolving accordingly. So what progress has been made in finding this clock. So from our side, we are we are not focused on the clocks themselves. We are like focused more on the connections between immunity and aging. This is because I'm a virologist by training, so I think this is the spot where I can contribute most efficiently. But I believe that the clocks can be telomeres, and this can be selfish evolution of mitochondria and maybe transposons. So these are three ideas that are like laying on the surface.
And I might add that, like judging from the fact that you said yourself, hey, will it look different? It's kind of easy to say from just the look of the person, how old this person is. So it might be in the extracellular matrix. Our aging clock might be there, because I mean, our surface is pretty much reliable on the state of extracellular matrix, and AGIs are being collected. So might be somewhere there, but this is just like a wild theory. Never tested. Yes. No, I mean, yeah.
Yes, why not?
Anyone else? Because we also have one more question from YouTube, but I would pass on to people here. No. Okay, next one from the YouTube then. How come the record-holding long-living vertebrates, Greenland shark and Greenland whale, are both Greenland? Is it connected to their ecology?
Well, it is tempting to connect, so I'm not sure it will be very reliable. My idea. So if they are like living in a abiotic environment, then they indeed might have less exposure, like let's say from spillovers from the pathogens from other species. Another factor can be just very basic, that they are living in a poor environment and they just don't have enough resources to reproduce like efficiently, and that's why they evolved longer lifespan as an energy saving strategy. So these are two ideas that I can pitch.
Anyone else, guys? Because my major question would be, what do we do about it? Like, do we try to tackle the immune system, or it will only, you know, and we'll end up dying from pathogens, or do we try to, you know, sterilize the system around us? Like, how do we try to fix this?
Anastasia, the system around us is pretty much sterilized. So because of our extreme longevity is probably explained that due to our brains, we can control most of the diseases. So that we if you think how much of our reflexes are about avoiding pathogens, it's amazing, right? So we avoid like feces and we avoid people who look strange because they are probably infected. And so a lot of stuff that we do, like non-consciously, is about infectious diseases. If you look in chimps, they spend most of their time taking out the ectoparasites. So we don't have fur probably to save time on that. So what we can do as scientists, I believe that if our model is correct, then gerontology is basically part of immunology. And this is a little bit of a shift, because now if I go, let's say, to Drosophila conference, there have nutrition, metabolism, aging section. And if my model is right, then it should be infection, immunity, aging. And this is a big shift. So the idea of that the idea that we can like use as to instruct our experiments that the connections between regular immune system and aging should be very, very intense. And that's what we use. So we believe that senescent cells are a primary immune adaptation, and we study acceleration of aging by infection. Like if the model predicts that if we are chronically infected and our immune system detects it, we want to accelerate aging. And this is what happens in HIV people, let's say. And we can model this in small animals. So there are some questions in the chat. Could you read them, please?
Yes, just a second. Sure. If this is so complicated or close to impossible after some time to evolve the lifespan, how do species do that?
I think the premise is wrong. It is not impossible or difficult to evolve lifespan because, for example, people evolved lifespan within like last 7 million years, like almost twice. If we come, so this 7 million years is our disconnection with chimps. So we are living, I think, twice longer, and we are large mammal with already long generation time and small effective population size. So and still we evolved it quite rapidly. A long lifespan evolves every time when the big body is evolving, then the brain is evolving, and the flight is evolving. So large animals are in all clades of mammals, almost. So every clade of mammals has some centenarian species. No, maybe not centenarian, but at least long-lived.
Oh, by the way, the size. There's this like question on aging. Why do big dogs, large dogs, live shorter than small ones? Is there some explanation in your theory?
So my take on this is that there are two correlations, right? There is ecological correlation that bigger animal should live longer because I cannot be a whale and live for 2 days. I need time to eat food, grow big, and use this size to propagate.
This investment.
Right? So I cannot afford a short lifespan. And this is a clear developmental constraint. So, but when we go to the individual species, like if we have a mutant that growth defective in flies, in dogs, in people, Laron syndrome, dwarfism, they have later onset of age-related diseases, at least if not the longer lifespan. So then we see a lifespan extension in these smaller mutants, and in mice, of course, the classical experiments. And this is probably associated with the growth. So, like, if probably it is a similar mechanism of lifespan extension as caloric restriction. If we see that we grow less, it might mean that the nutritional situation is bad. Then they expect to have less progeny, and then we want to extend our lifespan because then because all the models of this population dynamics is based on reproduction. So if we slow down reproduction, then selection slows down, and then we don't need to die as fast. So, and we want to give us time to reproduce generally.
Can I ask a question?
Sure.
Okay. First, only a few diseases lead to sterility, isn't it? So why why do we consider them? Maybe in some species there aren't any diseases which lead to sterility, and moreover, it's not the disease that lead to sterility have no profit to disease, to infectious agent, to
yeah, infectious agent. Yeah, I got your question. So the question is that we believe that not all the pathogens, of course, are selective factors for aging. So like influenza or common cold are not factors that select. So, however, like a classic evolutionary parasitology, it says that like a pathogen to reproduce needs some resources. These resources are taken from the host. Host is by definition less fit, and when it has a disease. The advanced pathogens are trying to take it from reproduction because reproduction is not that important for the pathogen. Host reproduction is not that important for the pathogen. And host survival is more important. So this is yeah, indeed, the question is very legitimate. So we specifically discuss it in our paper. In principle, if we have several pathogens that with low effects on host fecundity, then they can work the same way as a strongly sterilizing pathogen. Because let's say in
okay, so I can find a very complicated equilibrium in all these processes. If pathogen lead to sterility or not, or so. So I think that we should find some organisms that that have a long lifespan, as голый землекоп. Sorry.
Nate Mulred.
Nate Mulred. So I wonder if it's so hard to become a long lifespan organism.
Well, I don't think it's hard, because also, you know, this classic Michel Rosé experiment that they evolved Drosophila, the flies. So they in several years they extended their lifespan experimentally very significantly. So the evolution of lifespan can go fast. So coming back to the first part of the question, so I think that no one is pathogen free. Pathogens are the strongest evolutionary force on Earth. So if you just look at genes that are interacting with viruses, they evolve like 3 to 6 times faster than anything else. So I don't think that we can really find like a species that is free of pathogens. So it would be difficult. So the concern, if the sterilization by pathogens is a universal universal something, and if the degree of this sterilization is sufficient, this is a legitimate concern, and this an answer that I cannot. This is a question I cannot answer. No, because for that I need to study all the biosphere. But I believe there are good scientific reasons to believe that there are sufficient pathogen pressure across all the species.
So, sorry, I have one more question. So we rarely can observe this pressure of parasites because more often we can observe pressure of lack of food or predators. So mice should potentially live long because they often undergo predators and lack of food. So this mechanism you consider about parasites maybe not effective of them. They don't need to construct their mechanism in their genome.
Yeah, so this goes to the second slide of the lecture that we discussed selection shadow. So the animals generally survive to the age of senescence in the wild, and this is a basic ecological fact. So the also like people in the field often still discuss the effects of extrinsic mortality on the evolution of aging, but often these discussions are wrong because extrinsic mortality per se should not affect the evolution of aging. I can give you like references to the demographers' papers that show that. The key names are there, Caswell, and also a nice review has been published by Murad Promislov and Silvertown. So the extrinsic mortality shouldn't be a large factor because the still deterministic extrinsic mortality, like in killifish, can be a factor. If everybody dies, only eggs survive, then aging indeed can be neutral. If mortality is stochastic, then it is a completely different mathematical model, and then it should not directly affect the aging. So it can affect, but it can both prolong or reduce lifespan evolutionary.
Thank you.
I was starting to think that the clock question that also was raised, if our lifespan is limited by our immune system, basically, then the clock has to be immune-based too. So it cannot be telomeres or AGIs in extracellular matrix. Senescent cells, yeah, maybe.
Well, it might be that extracellular matrix can play a role in the immune system this way, but
oh yeah, absolutely, because the level of information just raises with the accumulation of AGIs,
but I generally believe in senescent cells. So I think this is the what is laying on the surface. So as you might know, this if we look in this cellular senescence, specifically in this SASP, senescence associated secretory phenotype, then we find that it consists more or less of innate immunity pathways, and that also P53 key regulator there it plays a role in the interferon pathway. And if you mutagenize P53, then you have slight increase in virus reproduction. And the P53 itself has been found by virologists, as you might know, so as an interactor with the T antigen of SV40. So interaction between senescence and viruses is known for a long time. So we are now studying the antiviral functions of senescent cells, and we have reasonable results that showing that indeed, if cells are senescent, they are more resistant to viruses, and they can protect healthy bystander cells from virus infections. So now our focus is on this direction, but it doesn't mean that it's the only thing.
Okay, another comment in the comment section. The problem is that to the moment, exactly none of approaches used for animals was effective for humans. Well,
yeah, this is
kind of hard to
this
test
is
on humans too.
yeah, but generally it is a legitimate claim. So, and I think we can, based on our model, we can explain why it is so. So it's because if we, let's say, we have two ecological factors. First, I need to reproduce. I need time for that. Second, I want to kill myself because of the infections, right? So let's say I'm a short-lived animal, like most of the experimental systems are short-lived because of the practicality. You need to be done by time. Then if, let's say, my reproductive period came to the bad weekend, which is rainy, then I'm screwed up. Then if the environment is not permissive for reproduction, I want to extend my lifespan. And this why smaller the animal is, shorter lifespan is, larger these hermetic responses we have. So in higher mammals, like in humans, which are very long-lived, we live long enough to have a shiny weekend, at least one, to reproduce. So, and with this, we don't need this much of this plasticity of lifespan to assist our evolutionary success. And that's why some of the approaches used for animals successfully could target this inbuilt plasticity that is existing and specific only in small, short-lived animals. And these mechanisms might be not around in us. And this is a difference for transitioning even from mice to humans.
You're saying your theory could be applicable to short-lived animals and to most of the animals, but not necessarily to humans, or not in full?
No. It's applicable to everyone, but we speak about plasticity in lifespan, right? If we have caloric restriction, let's say, then if you take worms, the effects are enormous. If we take mice, effects are much smaller. If we take humans, it's not known if the effect is there in the end. So the explanation for that can be produced if just a worm, if the weather is good during this, isn't good during these 3 days, the worm replicates, then the worm is screw up. So if conditions are bad, it wants to extend its lifespan. For us, we live for years, and during these years, we will find we are much less dependent on the oscillation in the environment. And that's why our lifespan is much less plastic. And if we find a drug which affects this lifespan due to this plasticity, then it will work in worm and not work with us. Okay.
I think someone else wanted to ask the question because I saw it. So people... Yeah.
Yes. So yeah. Sorry. Yeah. So I can join on the now. So I have a question. Okay. So the first thing I have a comment about on calorie restriction is like the normal thing that like it's a common thing. We say that okay. So we have like long, short-lived animals. We apply calorie restriction. We have like huge increase in the lifespan. And how to say so? And when we're applying the calorie restriction to the animals with a long lifespan, we have like less effect. But the thing is also that so when we take more long-lived animals, we apply not severe calorie restriction, because in mice, so the best results I achieved with calorie restriction, if I remember correctly, like 40%, and I don't believe that the same extreme calorie restriction was applied to the apes, because like with the 40% calorie restriction, they will feel themselves like very badly. So like because like there was after the Second World War, there was an experiment with their soldiers. So they were applied for something like they were subjected for something like 30% or 40% caloric restriction, like to mimic so the condition the prisoners of war faced, like in the Japanese, like in the Japanese camps, and it was like extremely harmful. And I believe that so we should also include in this logic that the more, how to say, the more complicated is the animal, so the more severe consequences we will have, with the caloric restriction. So maybe this also, one of the things why we don't see like, big effects like in the apps. But my question is so about the so maybe someone already asked it. So we know that clearance of senescent cells does not increase the lifespan of the mice like 2 times, 3 times. It gives like the effect that is even less than the caloric restriction. So do you believe that there are other mechanisms that also underlies the aging, and we need some combination approach? So we need to target senescent cells and maybe some other mechanisms, because it looks like that eliminating senescent cells only doesn't give like doesn't eliminate the aging.
Yeah, this is a question I don't know the answer. So I believe that senescent cells is a good direction, but I cannot comment on the exact exact progress on the lifespan extension, the status of the field. I don't think that senescent cells are understood well. And when you say that the elimination of senescent cells in mice, so how good this, how well this experiment has been done, I think is still might be not clear for. So if so you probably mean this P16 timisolate kinase constructs. But yeah, so Stas, this is a question I don't have an answer. I think we need to understand the system more. I believe the field is only, in my opinion, the field is very, very immature, and the insuccess in producing the therapy or intervention is not a main criteria for the priorities of choice, the directions.
May also expand on this question. So do you believe that we don't have like good markers of cell senescence because P21 and P16, so they are not these kinases, they generally play a role in the cell cycle progression, and they could be activated, inactivated, in many other conditions besides cell senescence. And actually so the way we define the cell senescence is combination of P16 expression, P21 expression, some staining for X beta gal, which I don't understand.
Also around the macrophages. Yeah,
it's marker, but we don't have any ideas if it's have some mechanical role. And we also look that the cells are prone to apoptosis, and these
resistant to apoptosis, right?
No. Yes, resistant to apoptosis. Yeah, and if we take P21 alone, or we take the resistant to apoptosis alone, it doesn't say that it's cell senescence. So we taking several unspecific phenotypes, combine them together, says, oh, this is senescent cells. So do you believe that maybe we need to find some more specific marker, or maybe some more specific signaling pathways that really will allow us very specifically distinct senescent cells from other states? Because at this point, I have some feeling that senescent cell is like some kind of like generic term. It's not like very specifically described term.
Yes. You just most people in the field will say exactly the same as you said. So, and I also agree that there is no clear marker for senescence, and every marker has like its off targets. So in combination of markers works to some extent, but we still don't understand much. And yes, senescence is not like a state like here you are senescent and here you are not senescent. Senescent is not binary, and there is more like a continuum of events. So some factors go up, then go down, then go up again. So NF kappa B, for example, it first it goes up and it goes down. Go up again. So during the transformation, it's a very strange phenomenon. It's a big thing, I think. So a big complex story, and I don't think that we understand it good enough.
So how many people in the field also share your vision and your, well, theory of aging and look at it from your perspective as well?
Very few. No,
Very
I'm like a pariah. So they the problem is that if you say that aging is adaptive, then you already posit yourself as a heretic. And because the arguments about Hamilton's rule and lack of the mortal mutants are very, very strong, and people discussed it like decades ago, came to a conclusion it cannot be adaptive. Then they remember this conclusion, but they have forgot which arguments they come to. And now our model is communicating with these arguments, but the arguments themselves are a little bit forgotten, and the conclusion is dominating. And that's why many people, when they just see the adaptive aging, this is a freak. And also, of course, that I'm Russian plays against me, because Russians have this Russian scientists looks in the clouds, thinking about problems, and this is also.
But you're in UCSF. You're not like too Russian.
No, right? But yes, unfortunately, my accent and other traits but just now this is a very marginal model, and it's difficult to get grants, difficult to even get talks in the conferences, and difficult to publish papers. Yes.
You published several, right? Like 2 or 3 on the topic. Yeah. Just pretty recently, so it's still new. We can still give it some time.
Yeah. So the first paper took 3 years to publish. So to
publish for the journals to recognize the paper itself. You mean?
Yeah. No, we were trying to iterate, reject. Finally, actually we got to PNAS, and just by protection of Eugene Koonin. And then starting from there, so I found that ecologists are much more receptive to the crazy models, so that the gerontologists just ignore. Ecologists are excited because if this model is correct, this is like a founding model of gerontology, and it is one of the key discoveries in ecology, because they have also this life history theory, which is evolution of aging related. And there, trends of ecology evolution, they got enthusiastic about the paper, published it without major efforts from big shots. Yeah.
Anything new cooking right now? Anything in the making? Any
Oh,
new
yeah,
article?
Yeah. We have one in writing and one model and experiments justifying the models. In flies, we have this cellular cell senescence, which is still immature, but we got very strong effects in virus cell culture. We have couple more theoretical papers under construction, like why similarity evolved, like just modeling and fitting field data, rejuvenation. We can prove their theorem. So the rejuvenation is a major flaw in the not just the this mainstream models that are proposed by Charlesworth and other classic gerontologists. They presume that biological age monotonously depends on time, and this is like an implicit assumption of their model. And since we see rejuvenation, it's clearly not true, and we can just disprove them. It looks like, so it's still under construction. It looks like that we can disprove them just mathematically. Yeah, so we have a lot of stuff to do. If you want to collaborate, we're happy to. If you have funds to help us, we will be more than happy to take them.
I think it's a fascinating approach. I think it's super interesting. We'd be happy to be involved. I'm not sure how at this moment. Let me think. Let me look into what's happening right now. But yeah, I encourage everyone, if you have ideas, to be involved. I think it's frankly we don't have it solved yet. Like Peter is saying, hey, everyone is looking at me as I'm like a weirdo with this new theory. But I mean, the old theories are not justified. It's not like we know what aging is. It's not how we figured it out. So why not look at it from different perspective, like from like a new bold perspective? Yeah, why not?
Yeah, just the most of the just a large proportion of people in the field think that they need to find druggable targets, and they don't care about what aging is. And this is like this Antikythera example. Then you are trying to follow the molecules of decaying brass instead of focusing on the whole picture. I think this is a wrong approach that we need to because questions that why aging exists is probably simpler than the question of what happens during aging. And I think we first need to solve simple questions and then go to get instructed to solve complex questions. And this position is, that's how science works and medicine works. Like you first set up the diagnosis, then you start treatments. But this system of value somehow is not shared by many researchers in the field that think that just trying different molecules or combinations or different regimes of interventions, they can improve situation. But to me, it is like I need to emphasize this with Stanislav. We have like a long heated discussion on the Telegram about the approaches and about combinational therapies. So that the argument that I produced that before the germline theory was invented, there were so the diseases are caused by microbes. So there were a lot of other models. Let's say there was a model of the miasma, that the bad air is bad, or there was like a model of bad eye. And based on these models, we can invent other interventions, right? Like ventilation or hygiene for miasmas, or like hijabs and burqas for bad eye. And both ventilation and heat jobs would work to some extent, because ventilation will remove pathogens from the air, and the heat jobs will work like masks. And endorsed by these small results, researchers can say, okay, now we will do super ventilation or super heat job. That will solve problem entirely. But all these
or use them in combination.
Or use them in combination. Yes, heat job with ventilation. That was our yes. But the just encouraging results that seem to be minor, but encouraging might be completely misleading. And without like a paradigm in the field made, we cannot prioritize that. We cannot evaluate the perspectives of certain intervention.
So, by the way, do you know of any current tracks of drug development in the field are absolutely misleading and wouldn't work, given that your theory is right?
Drugs?
Yeah, I mean, like some companies are trying to develop some anti-aging drugs, some therapies. Like,
no, so my belief is,
yeah.
No, I don't think that our model can be proved based on drugs.
No, can be. I mean, no, I'm asking a bit different.
Okay,
so
like, who
companies are trying to find therapies. No, we're not to invest, or like, I don't know, maybe not names, but just approaches. Like, do you see any obvious contradictions between your theory and current state of affairs in the field?
No, so the as I said before, I believe senolytics in general should work. So and the major Tom Kirkwood, who is the author of Disposable Soma, he is publishing paper after paper that senolytics shouldn't work, because if the senescent cells evolved, they should be good for something. If we remove senescent cells, then we will probably harm the organisms, so the senolytics should have limited effects. So this is probably the main contradiction, but yeah, I also believe that caloric restriction and this type of things, they might be of limited use for humans because of the yeah, but I'm not a big, so I'm more like a fundamental scientist. I'm not aware of most of the development of drugs.
Alexander is adding here in the comments that the most profitable is to be long lifespan in one persons with short lifespan.
Yes, yes, it's the best one to be an egoist and surrounded by altruists. Yes, this is a classic game theory. Hi, Alexander.
I had a name of my account, and now I should better to rename myself. And as we talk about drugs, should we? How do you feel about physical training for lifespan? What do you think about it?
Physical
training. Physical.
Yeah, I got. So this is the only I think one of the few and the strongest intervention that is shown to indeed improve the lifespan and life quality. So how does it is it explained by our model? I don't know yet. So I think just in nature, most of the animals are like exercising continuously to find food, and why the decline in physical activity should shorten lifespan, I don't know.
Moreover, in nature, most of animals avoid physical activity. It's very good to lie down and relax.
Yeah, but because they have hard work all the time, right? And they just have to rest. But we don't have as much physical activity as is assumed by our evolutionary design in our day-to-day life, and we sit in the front of computer writing grants or stuff. So that I think our lifestyle is completely artificial. So probably if we put cats or dogs in the position that they can sleep the entire day, they will also be not very happy in terms of lifespan about that.
But if we say about home cats, they have enough food, and they live. I don't think that they live shorter than wild one. Cat lives about 20 years, isn't it?
Yeah. No, I'm not sure this is like a fair experiment. So you need to compare really sedentary cats and actively moving cats. So no, this is a question I cannot answer. I think physical activity is improving health in most species. So in flies and in mice, if you put them a wheel, then they live longer. Maybe they exercise. So yeah. So I'm not sure about pets, but the physical activity is good is also shown on laboratory models. Yeah. I think Stanislav probably wants to add on the physical activity.
Yeah, there is like amazing data. So there is such a thing as ischemic preconditioning when you apply the short terms of the ischemia. So before the longest ischemia, and it protects heart, it protects kidney, it protects it protects brain, it reduces the ischemic damage in many tissues. And then it was found out that you can also do remote ischemic preconditioning. For example, you apply short ischemia to the hand or to the leg, and this induced signaling that protects again myocardial infarction, against stroke. And when this remote ischemic preconditioning was discovered, so it became clear that it could be translational. And then people performed so there was several clinical trials in humans that it was found out that so the older is individual, so the less effect of remote ischemic preconditioning we have. But if these if you compare older subjects who has their low level of physical activity and high level of physical activity, so those with a high level of physical activity, so the effectiveness of the remote ischemic preconditioning becomes much higher, and this real world evidence showing that that physical activity is really very beneficial, in slowing, in contracting, so the effects of aging. So I think it's very difficult to imagine that somehow the physical activity will be damaging to humans. And I would say that in the wild nature, so animals are resting because they don't know so when they will need to run, because they live in the constant fear of being attacked. And so how to say, you can't afford so people can go to the gym and can afford to be exhausted after gym, so because you don't expect that you will leave the gym and you will have run for your life. But animals they always expect run for their lives. And so when they will have when they have time to rest, they will rest, because they don't know for how long time they can rest. So I think that's why they have some kind of behavior.
Yeah, I think it's similar to what I said. Yeah, thank you, Stas. Can you drop me this paper on the intervention? Dyskinesia? Yeah, I haven't read it.
Yeah, I will try to find it. Yeah.
Thank you. Okay. Any more questions?
just wanted to add that we also had some of the guys dropping the article in the comments, and it was common features of agent fail to occur in race without a bacterial microbiome. So yeah, probably something interesting to look at. Yeah, you can just look it up. The link is in our comments. Probably Danilo will save it, or I can just send you the link later.
Send it to me.
Yeah, sure. And also we were mentioning that Peter had a heated discussion with Stanislas here, and like some other folks were having the discussion on the same topic that we have a lecture on today. If you want to join these heated discussions, it's mostly in Russian, but it's in our AGCT Telegram forum and some others too. I think we'll just leave a link in the description of the YouTube video to just join there. We have semi-friendly environment there, but feel free to leave comments.
We have explicit academic culture in Russia. So if Stanislav tell me that I'm an idiot, I don't get upset. We're still best friends.
Okay, can I?
Yeah.
Okay, so if anyone doesn't have questions, may ask one more. So I totally agree. Like in Russia, we have very toxic tradition of making discussions, and like when I moved initially to Germany and then to United States, it was like very surprising to me that you give a talk on the lab meeting, and your PI starts, "Oh, it's amazing work," but because like in Russia, so when you give like a talk on the lab meeting, your PI says, "Oh, this is wrong, this is wrong, this is wrong, this is wrong," and says, "But yeah, it's a good work," So there. Okay.
You need to give a classic bullshit sandwich. Something good, some critiques, and something good again.
Yeah. Okay. So, but
my better.
My question was Peter said that okay, so we need to develop a paradigm, how to say it in English, paradigm. And then we will understand, which targets, are druggable, which targets are not druggable. But how to say, so the question is, if there is some exact plans of, so which experiments, or I don't know what type of bioinformatic, work we should, do, and, to get, these results. And so if you have, the idea of of exact experiments, so how long you will think it will take to understand which targets according to which targets according to your hypothesis are druggable and not druggable, because so if it will take decades to identify the druggable targets, there is a possibility that maybe using our wrong unscientific approaches, we can find still the best targets faster. So what timeline do you expect?
Yes, I think the premise is wrong. I don't believe that just by random search, like Monte Carlo, looking a little bit here, one can find a solution to the most complex health problem that humanity has ever encountered. So just, the smallpox vaccine, right? It was found by chance. However, technical, because the people found that these women who are taking care of cows, they typically don't get sick with smallpox. They found the cowpox and start propagating. However, technical opportunity to do that we have had for 10,000 years. Right? For making a smallpox vaccine, we need a cow and a sharp stick. That's it. So, however, it took us 10,000 years to find the vaccine with Monte Carlo method. So the only way how to do things efficiently is by rational, scientific, iterative scientific approach. So this is my principal position. So the still is there an opportunity to find like a drug with the random search, like 1 million molecules? Yes, there is. How big is it? I don't know, because I don't know what are we finding. Just think about vaccinology. Would we find vaccines with the 1 million drug challenge, like by testing many drugs in worms or in yeast or whatever? No, we will not. So because the idea that we take a sick person, we take materials from the sick person, we somehow perturb them, and then we put them back in the healthy people, is not something that can come into mind of the person who thinks that diseases because of the bad eye or of the bad air, right? So the just the solution for aging might be not in a small molecule, or not in that small molecule that we think about, and without like a clear-cut idea what's going on, we might not see that. Well, technologically, we might be ready right now for this, like in the examples with the vaccines and cows.
It's difficult for me to agree, because like right now, so like diabetes community, so I'm working in, so it's like very encouraged with like two examples. So one example is inhibitors of sodium glucose transporter as SGLT2 inhibitors, and another is the agonist of GLP-1 receptors, semaglutide, or more famous like Ozempic, the brand name. And like, you know, the story of inhibitors of SGLT2, like amazing. So like the idea was like, you know, okay, so we have like, you know, so we have the channel that reabsorbs glucose in the kidney, and if we block it, so the person will start lose the glucose, and well, with the urine, the blood glucose will down, and it will be beneficial with patients with type 2 diabetes. And how this drug, you know, was developed. But when the clinical studies started, it was found out that so the the level of so the decrease of risk of cardiovascular events, the decrease of risk in chronic kidney disease development is you know, much higher than the effect of like lowering glucose. And then there was like additional clinical trials when the inhibitors of SGLT2 were done in the people without diabetes, and wow, so like, you know, you really see that even without the diabetes, you also have the decrease in cardiovascular disease, in chronic kidney disease. And so like, you know, there recently it was found that surprisingly, so inhibitor of SGLT2, they are decreasing the risk of cardiovascular events in older people, so with a higher efficiency than in the more younger people. And so like, you know, recently, so like the like previous weekends, I was in the conference, and there was a study showing that SGLT2 inhibitors, they promotes the clearance of senescent cells and so on. And so like, you know, so and like, you know, the second thing also, like with the agonist of GLP-1 receptor, because like the idea was like, you know, they're easy. So GLP receptor promotes more insulin secretion. It was also like developed, the drug developed as for treat diabetes, but then it was found out that it promotes the weight loss. And it was found that these receptors are also expressed in the brain. That's why, you know, it has this effect. And now there was a trial where when they're just, you know, obese people were on the Ozempic, and so the researchers were looking for incidence of cardiovascular
Story gets
events.
long, Stanislav.
Yeah. And so it was found out that, you know, the decrease in the cardiovascular events happens before the patients lose weight. And so like, you know, so like, you know, the random drug discovery, like it's the two examples when you just, you know, made, you know, just, you know, some random, you know, drug discovery, and you found that, you know, it has like, you know, lots of target, but they like there are beneficial effects. And that's why, you know, I don't believe like, you know, that like without so that Monte Carlo method, as you said, is so inefficient that we wouldn't be able, to find the way to treat the aging.
Yeah, well, Stas, this was not a Monte Carlo method, right? They targeted a specific protein that they targeted it. They found that it is very good to target it in the context of many, many diseases, right? So in case of aging, we don't know what to target. That's a problem. This is first. Second, I'm not that ambitious to say that people, you are doing something, don't do that. Do this, but I don't believe that your way is shorter than a regular way science was performing for the last couple hundred years. Paradigm, testing the paradigm, conclusions of the paradigm, building upon the paradigm. So the can be that someone tomorrow will develop a pill against aging that will solve all the problem. It can be. The probability is non-zero. But I believe that the most conservative and the most efficient strategy is to do it like it has been done for many years. So I understand the all under pressure because we all age, and it's not a big joy to see your parents dead, right? But still, we don't need our emotions to affect us that we are giving away the time-tested classic rational approach. At least in part, we can pursue this strategy. This is my strong belief.
I propose we slowly but gradually go towards.
Yes.
Especially given that Peter has given us this fantastic talk. We asked some questions. The only sad part about it is we actually have a youth group here, and they were shy, not asking any questions. So guys, I encourage you to maybe try to find another way. If you don't feel comfortable, ask here on the call. Let's maybe make a group chat, another one, maybe two in Russian English, because mostly the group is English speaking. We can do both. Probably written language could be easier to communicate with each other. So it's actually that we have such a big and active group of young people. We need more energy, as some people say in our transhumanistic groups. I don't know, Peter, any two last words? No?
Yeah, thank you very much for coming. Thank you very much for questions. It was exciting. And let's continue that. So I'm around on all the social media, and if you come up with questions, ideas, I will be happy to respond.
Thank you, thank you, Peter. This was amazing. Thank you, guys. See you around. See you in our next meeting.